Hello, ladies and gentlemen. Welcome back to my YouTube channel. So today I prepared for you something on histology. So, histology, basically, we are going to look on the immunohistochemistry of the breast cancer tissue. So, as a medic or as a young medical doctor, you can be requested now to prepare or to ensure that the tissue presented to you in a histological lab is fully processed and is ready to be examined either under an electron microscope or either on a light microscope or either on a PCR during the detection of the antibodies, or even on the lasers machines. So basically we are going to look here on the immunohistochemistry staining technique. So this is a very valuable tool that has been used to detect the presence of the antigens that determines whether cancer is there or not in the breast tissue. And actually, on my area, I'm going to actually specialize in the deparaffinization of the breast cancer tissues presented to you in a histological lab. So it basically entails several steps to be taken in order to accurately determine this experiment. So the first one here is the slide preparation. That is the first step. The second step under immunohistochemistry of breast cancer is the preparation of the primary antibody and its reaction. The third one is the secondary antibody reaction. The fourth one is the preparation of the substrate. The fifth one is the development of this substrate now. And the seventh one, is the counter staining. The eighth one is the mounting. And lastly, the microscopy and the results. Yeah. So we are going to start with the slide preparation. And actually earlier there I've told you that we are going now to deal with our deparaffinized breast tissue. So deparaffinized breast tissue actually is a tissue that has been embedded initially onto our tissue gazette, have undergone microtomy, and now we want to remove this paraffin. So how are we going to remove this paraffin now from this breast tissue sticked onto the microscope slide? We are going to take now this slide, place it into the oven, close the oven, regulate the temperature at around 56 to 60 degrees Celsius to melt off the excess wax. And then after that, remove it and cool it at around 20 minutes, at around 18 to 25 degrees Celsius, that is room temperature. Then after that, now you can use xylene. So xylene here we are going to use is to ensure that the excess paraffin has been done away with. So that is the first step under slide preparation. After this, after ensuring this has occurred, we are going to use the hydration of alcohol, the ascending graded of alcohol from 90% to 80% to 75 as you go downwards. So this one is to ensure that the subsequent procedures are especially not tampered with during the immunohistochemistry staining procedure technique. So that was the first thing under slide preparation. The second thing under slide preparation here, we're going to unmask the antigen, unmasking the antigen or antigen retrieval. So the most majorly used here is the microwave antigen retrievals. The microwave antigen retrieval is actually the mostly used method because it is cheap and reliable. So on the microwave antigen retrieval, the first step you're going here to see is one, you take the microscope slide now, which is been sticked, the breast tissue has been sticked into the microscope slide. You take that slide, you dip it into the microwave resistant jar containing that retrieval solution, and you ensure during this procedure that the slide is actually submerged completely onto the jar. Close the microwave and then you incubate it at around 56 to 60 degrees Celsius for around 20 minutes. Yeah. And then you ensure that you do a repetition around 2 to 3 times of this procedure. So this basically means you're going to do away with the first solution, the retrieval solution of the antigen, and you add another one until all the antigen have been unmasked. Yeah, so that is the main procedure in the microwave antigen retrieval. The third step under the slide preparation we are going to see is on the hydrogen peroxidases, that is the endogenous peroxidases. We want to remove them. The endogenous peroxidases basically will going to react to the our antigen we want to see on the breast tissue. So we want to do away with them. And how are you going to do away with them? The first step we're going to see here is take the microscope slide, place it on a rack, yeah, ensure that it is flatly laying on a rack. Then the second procedure you're going to perform here is you take 3% of hydrogen peroxide solution. You ensure that the slide is fully submerged into that solution. Then you incubate for around 5 minutes. After that, you wash, then you rinse, and then you tip off the excess water, and then you dry it, waiting for the next step. So that one basically was the first step on the slide preparation. The second one is the primary antibody reaction. So the primary antibody reaction basically here entails the use of antibody, the primary antibody, to react with the weak antigen that may be found on our breast tissue. We don't want any weak antigen to be there. So the first step here we're going to see is the primary antibody reaction. So the primary antibody reaction here, we are going to take our microscope slide, we place it on a rack. After that, we're going to take our primary antibody, which is a dilution. We then dilute it up to an optimum level in a diluent. And then now you take around 100 microliter of that the diluted antibody, and then you submerge it, yeah, onto the microscope slide. Incubate it, yeah, for around 60 minutes at 37 degrees Celsius actually. And then after that, you wash, you rinse it, and you clean. You ensure that it is dry, waiting for the third step. So the third step here is the secondary antibody reaction. So the secondary antibody reaction here, especially we are going to use the biotin or the extravagant solution. The biotin or the extravagant solution is also going to be diluted up to optimum dilution using a diluent. After that, we are going to take our tissue, stick it onto the microscope slide, place it on a rack, and then you take a drop of that secondary antibody and ensure that the slide is fully covered with the antibody. After that, we are going to incubate it also, but for in this time you're going to incubate it for around 30 minutes, yeah, at around 37 degrees Celsius in an incubator. Then after that, at the end of that procedure, we are going then again to wash using a distilled water. After washing using a distilled water, you are going to tip off the excess water, yeah, on either a gauze, and then after that, you are going to dry our microscope slide, waiting for the next procedure. So the next procedure actually here is the substrate preparation. The substrate preparation, we are going to prepare our substrate using basically 1 molar levamisole, then diluted it in a phosphate solution to prepare our substrate. So basically that one you have prepared your substrate solution. Then the next step here is the substrate development. So substrate development here, we are going to take our microscope slide, place it on a rack, yeah. Then you take 1 drop of that substrate prepared solution, then you cover it uniformly. Ensure that the whole microscope slide is entirely covered with that substrate solution. Yeah. After that, you incubate it for several minutes, actually 0.5 to around 5 minutes. Yeah, for about 37 degrees Celsius. So you ensure that you give it time for the substrate solution to actually react to the antigens on the breast tissue to ensure that a synergistic color has been gained and has been absorbed. And how are you going to look at this? Or how are we going to determine that our color of preference has been attained? Take the slide, place it under a light microscope using a preferable power objective lens. You are going to determine that the endpoint actually has been attained when you now see the color you wanted to achieve. So terminate the reaction after that using distilled water. Wash the slide, yeah, to remove now the surface solution off, because you don't want to understain or overstain the background staining. Yeah, so this one basically will help us to achieve the vision when you want to observe it either under light microscope or under electron microscope. So after that, you wash it with water, tip off the excess water, then you dry it on that gauze. You wait for the next procedure. So the next procedure basically here is the counter staining procedure. The counter staining procedure, the mostly used stain here is the H&A, that is the eosin and hematoxylin. So in this case, we're going to use the Meyer's hematoxylin. The Meyer's hematoxylin here is preferred because it is majorly used in exfoliative cytology and even in cytology in the study of cancers, in the study of cervical cancers, that is the exfoliative cytology, and even in the breast tissue cytology. So we are going to use the Meyer's hematoxylin, where now you take the prepared slide, place it on that rack, take the Meyer hematoxylin, and then ensure that it is fully covered. Yeah, it is fully covered in Meyer's hematoxylin. Incubate that microscope slide after doing that for around 20 minutes. Then after that, you're going now to wash again using distilled water, tip off the excess water and dry it as it waits for the next procedure. So the next procedure is basically mounting. So mounting here, we're going to use the known solution, that is the DPX solution, to mount it. You can also use the ring media around the cover slip of that microscope slide, yeah, to ensure that the air bubbles has actually been prevented. After that, we are now going to mount, yeah. We are now going to do microscopy. Microscopy here, now we can either use the electron microscope or the light microscope under a preferable high power objective lens. So in the electron microscope, you're going to use the metallic stains to achieve several structures. We can either also use even the lead hematoxylin, yeah. We can also use the molybdenum hematoxylins in the electron microscope, because actually electron microscope deals with the staining of the heavy metals. Yeah, so after that, it is going now to be pictured in the either in the computer, and then it's going to be printed as a micrograph, which is the final result that is going to be examined. So under microscope, actually now our results, we want to see if the tissue actually is having a cancer, a possible malignancy, which can either be a carcinoma, yeah, or can be a sarcoma, yeah, or it is a benign tissue that is actually undergoing a progression to become a possible breast cancer. So how are you going to do? How are you going to determine that actually this one is a breast cancer? So breast cancer, actually immunohistochemistry of breast cancer is basically determined by the markers, what we call markers. So after substrate reaction, there will be markers now. We are going to determine this by several markers. So the first marker is the estrogen marker. The first one is the estrogen marker, abbreviated as the ER. The second one is the progesterone marker, abbreviated as the PR. The third one is the human epidermal growth receptor factor 2, abbreviated as the HER apostrophe 2 or a hyphen 2. And then you are going to look at the marker called the calponine marker. You are going to look at the marker called the CD10 marker. You are going to look also at the marker called the Ki67 marker, and we are going to look at lastly the P63 marker. Yeah, so let's start with the ER, or the estrogen receptor markers. So what does it basically mean? So overexpression on observation under either electron microscope. Yeah, because actually this is a virus. We're going to use an electron microscope. So ER receptor marker, actually the overexpression of that on that breast tissue actually will determine the tumor grade of that tissue. Yeah, it will actually tell us the predictive and the prognostic factors of that tumor grade on that breast tissue. The second one is the progesterone marker. The progesterone marker basically will depend on the estrogen marker actually. So it cannot be there without the estrogen receptor marker. The third one here is the HER marker, the human epidermal growth receptor factor 2. That's the HER2 marker. This one is going, so the overexpression of HER2 is basically means there is a proliferation index. Yeah, there is a carcinoma index. There is high carcinoma index, basically suggesting that there is a possible cancer in that breast tissue. The third one, the next you're going to look here is the calponin. Calponin basically will always deal with the contractile elements. So overexpression of the calponin, yeah, markers on that breast tissues when observed under electron microscope on the preferable high power objective lens will going to tell us that there is a possible malignancy, suggesting that there is a breast cancer because there is overexpression of the contractile elements on that breast tissue. The third one, the next we are going to look at is the CD10. The CD10, the overexpression of the CD10 actually will tell us the overexpression of the stromal myofibroblasts found on that breast tissue, thus suggesting there's a possible case of malignancy, that is breast cancer. Yeah, and another one we are going to look here is the Ki67, which also the overexpression of the Ki67 will basically mean there is high proliferation of those tissues, those basically proteins. So ensuring that actually this one is a possible malignancy, which will result into a cancer, basically the breast cancer tissue. Yeah, so that was basically how immunohistochemistry technology technique is done, and you do it up to the end where now we achieve our end results by discussing the markers, which will now either tell us are these possible causes of this breast cancer or are they not there? Is it a benign cancer, or is it a carcinoma, or it is a sarcoma? Yeah, so that was basically what I prepared for you. And next week we are going to look at more of pharmacology. So stay woke, my gentlemen and ladies. Until next time, and please subscribe to this channel. Thank you.